Hello everyone, welcome to my YouTube channel. If you are new to my channel, please subscribe, and I have a big appreciation for your kindness. Today I am going to discuss about cellular metabolism. Metabolism is a biochemistry. What is biochemistry? It is a chemistry within living organisms. When we say cellular metabolism, metabolism is sum of chemical reactions. All cellular activities require energy, therefore energy should be made by enzymes-mediated processes, that is metabolism, specifically catabolism, which is the process to make ATP. When you say energy, it is ATP. Metabolic pathway, when you say pathway, you can relate with the path through which you move somewhere. You move to your school, you move to your college, you move to your office, your town. Why you are moving is just to do something. Like that, metabolic pathway is just to do something. In this case, just to make what? ATP. What are the pathways? For example, glycolysis, TCA cycle, and others. And living cells are in constant activity. For example, macromolecules are assembled and broken down. These macromolecules assembled from smaller molecules, and when it is required, these macromolecules will be broken down. Substances are transported across cell membranes. Genetic instructions are transmitted. These are always there. They are constant activities and they need energy. Therefore, energy should be made. Metabolites, the reactants and products of chemical reactions are called metabolites. What are reactants? To the left side of the reaction, and what are the products? To the right side of the reactions. For example, A plus B yields C plus D. Here there will be an enzyme or enzymes. Therefore, these reactants and products are metabolites. Or A, in the conversion of A to B, A is reactant and B is product. Metabolites include proteins, carbohydrates, nucleotides, lipids, coenzymes, and cofactors. Enzymes are biological catalysts. They are within living organisms. They make a chemical reaction millions of times faster. This is very important. Almost all metabolic processes in the cell need enzymes. Enzymes speed up reactions by lowering activation energy. That means the requirement of energy to convert one reactant to product will be low if there are enzymes in the process. For example, in this graph, the y-axis indicates energy and the x-axis to the horizontal is enzyme action. Here, this line indicates if there is no enzyme, and this line indicates if there is enzyme. Therefore, to convert reactant to product to the same level, the energy requirement will be low if there are enzymes. This amount, the energy of from this level to this level. If there is no enzyme, the energy requirement will be from this level up to this. A lot of energy will be required. Therefore, enzymes save energy for cells. This energy will be utilized for other cellular activities. What is induced fit means? Induced fit is important for many enzymes. Enzymes change shape when substrates bind, meaning catalytic activity be induced by the binding of the substrate. That means the substrates are the alert for the enzyme's activity. When substrate come and contact with the enzyme, then the enzyme activity will start. Substrate is capable of inducing the proper alignment of the active site of the enzyme, causing the enzyme to subsequently perform its catalytic function. Here is induced fit action. This is an enzyme and this is a substrate. Then this substrate, when this substrate come and contact with this enzyme, then the process will begin. Therefore, these are the two different molecules, and after the process of enzymes catalysis, it will be converted into different molecules. When you look at this color and this color, which is different from this color, okay, these are the products. Chemical nature of enzymes. All known enzymes are proteins, with the exception of recently discovered RNA enzymes. The recent discoveries show that there are RNA enzymes, but we generally know that enzymes are proteins. Enzymes are high molecular weight compounds made up principally or mainly of chains of amino acids linked together by peptide bonds. We know that, or here it says, enzymes are proteins, and proteins are made of amino acids by peptide bonds. Two amino acids bond together by peptide bonds and finally form a protein. And enzymes are proteins. Therefore, the building blocks of enzymes are amino acids. Many enzymes require the presence of other compounds. These compounds are called cofactors before their catalytic activity can be exerted. Before the catalytic process of the enzymes, there should be attachment of these compounds or cofactors. This interactive complex, that is the enzyme plus cofactors, we call the complex. This is referred to as the hollow enzyme. Therefore, apoenzyme, that is protein portion, and the cofactor is the hollow enzyme. For example, in this picture, this the protein part, the larger part is apoenzyme, and here is coenzyme, this part, and this one is cofactor, and here is the active site or catalytic site where the substrate will come and attach it, and the whole complex we call hollow enzyme or holozyme. On the basis of differences in chemical nature, enzymes can be classified into simple enzymes, conjugate enzymes, metalloenzymes, and isoenzymes. Simple enzymes are made up of only protein polypeptide. They contain no chemical groups other than amino acid residues. Digestive enzymes such as pepsin and trypsin are of this nature. They are simple enzymes, only from amino acid molecules. Conjugate enzymes, these are apoenzymes, which we have seen before. The protein part and cofactor or non-protein part are there. They are also called hollow enzyme. There can be an enzymatic activity only when both components are present together. If there is no one component, there is no function of that enzyme. Both components should be there. The cofactor is sometimes a simple divalent metallic ion, such as calcium, magnesium, zinc, cobalt, etc., and sometimes a non-protein organic compound. If the cofactor is firmly bound to the apoenzyme, we call prosthetic group. If it is firmly bound there, we call prosthetic group. For example, cytochromes are the enzymes that possess porphyrins as their prosthetic groups. If instead of being more or less permanently bound to the apoenzyme, the cofactor attaches itself to the apoenzyme only at the time of reaction, it is called coenzyme. If they are always there, we call prosthetic group. If they come during the process of enzymes catalysis, we call coenzyme. These are not permanent. They are not permanently attached to the enzyme. The third class of enzymes are metalloenzymes if the metal forms part of the molecule, for example, as iron of hemoglobin or cytochrome. From the name metalloenzymes, metallo means for metal, and then metal plus enzyme we call metalloenzymes. The metal cofactors involved in enzymatic reactions are monovalent as potassium ion, divalent cations such as magnesium, manganese, and copper ions. They are loosely held by the enzyme. These are permanently there, but in this case, they are loosely held and they are not permanent. The fourth class is isoenzymes. Iso we can call also isozymes. A number of variants of an enzyme producing the same product. Iso means the same. Why it is called iso? Because a number of variants of an enzyme. One enzyme having different parts of or different kinds of enzymes. Variants of enzymes will make what? The same product. The enzymes are different, but they make what? All make the same product. Therefore, isoenzymes or isozymes. Then classification of enzymes. Class of enzymes based on the substrate they act upon. How we classify these enzymes are based on the substrates they act upon. Accordingly, there are 6 classes, the major classes. The first one is oxidation reduction. And the description is it transfers electrons. The enzyme class is called oxidoreductases. ASE indicates the enzyme. And group transfer of functional group from one molecule to another. This class of enzymes are called transferases. From the name, group transfer. Hydrolysis, cleavage of bonds by the addition of water. So the enzymes are hydrolases. Non-hydrolysis cleavage, cleaving or splitting molecule by non-hydrolysis process. That enzymes class are called Lyases. And isomerization and rearrangements. The description of the reaction is rearrangement of atoms to form isomers. The enzymes are isomerases. Ligation using energy from ATP. Ligation means joining. So formation of carbon and other bonds with energy from ATP, the enzyme class are called ligases. Mechanisms of enzyme action. An enzyme attracts substrates to its active site, catalyzes the chemical reaction by which products are formed, and then allows the products to dissociate or separate from the enzyme surface. This is very important property of enzymes, that the enzymes will not be consumed in the process. Okay, they will be finally free. They release the products and then they will be free. Then they will be ready for the next process. The combination formed by an enzyme and its substrate is called the enzyme substrate complex. For example, here is a substrate, S indicates substrate. This one is an enzyme. This site is active site. If this substrate come and attach with this enzyme, we call this enzyme substrate complex. This is the enzyme plus the substrate. The substrates are attracted to the active site by electrostatic and hydrophobic forces, which are called non-covalent bonds because they are physical. They are physical attractions and not chemical bonds. What is electrostatic force and hydrophobic forces? Electrostatic means relating to stationary. Okay, stationary, not stationary. Differentiate stationary. Here is A, but here is E. This is for the shops in which the papers and pens and other academic materials be sold. This one is stationary, means fixed. Therefore, relating to stationary electric charges or fields as opposed to electric currents. Electric currents are charged particles or ions or electrons at motion or movement. In this case, it is what? The electric forces per unit charge, and this is not referring to the movement. Therefore, the force is what? Electrostatic force. Electrostatic forces are non-contact forces. They are non-contact. They pull or push on objects without touching them. Factors affecting enzymatic activities. There are different factors which affect enzymatic activities, such as temperature, pH, that is acidity and basicity, substrate concentration and enzyme concentration. Let's see each of them. Increasing temperature increases the kinetic energy that molecules possess. Kinetic energy is the energy related with motion or movement, just relate with your physics knowledge. In a fluid, this means that there are more random collisions between molecules per unit time. Collision means movement. Since enzymes catalyze reactions by randomly colliding with substrate molecules, increasing temperature increases the rate of reaction, forming more product. When temperature increases, the collision increases, then there will be a reaction between the molecules and the enzymes. Therefore, there will be more product. However, increasing temperature also increases the vibrational energy that molecules have, specifically in this case enzyme molecules, which puts strain or damage on the bonds that hold them together. As temperature increases, more bonds, especially the weaker hydrogen and ionic bonds, will break as a result of this strain. Breaking bonds within the enzyme will cause the active site to change shape. This change in shape means that the active site is less complementary. Complementary means which can fit to the shape of the substrate, so that it is less likely to catalyze the reaction. Eventually, the enzyme will become denatured and will no longer function. Denature means destroyed. The temperature at which the maximum rate of reaction occurs is called the enzyme's optimum temperature. This is different for different enzymes. So, different enzymes have their own optimum temperature. The other factor is pH, that is measure of acidity and basicity. pH is a measure of the hydrogen ion concentration and therefore a good indicator of the hydroxide ion concentration. Lower pH values mean higher hydrogen ion concentrations and lower hydroxide ion concentrations. Since pH means power of hydrogen, it is measuring the hydrogen ion concentration. Hydrogen ion and hydroxide ions are charged. They are charged, positively and negatively charged, and therefore interfere with hydrogen and ionic bonds that hold together an enzyme, since they will be attracted or repelled by the charges created by the bonds. This interference causes a change in the shape of the enzyme and, importantly, its active site. Different enzymes have different optimum pH values, like the different enzymes have their own optimum temperature. The same is true for the pH. This is the pH value at which the bonds within them are influenced by hydrogen ion and hydroxide ions in such a way that the shape of their active site is the most complementary to the shape of their substrate. At the optimum pH, the rate of reaction is at an optimum. Any change in pH above or below the optimum will quickly cause a decrease in the rate of reaction, since more of the enzyme molecules will have active sites whose shapes are not, or at least are less complementary to the shape of the substrate. Small changes in pH above or below the optimum do not cause a permanent change to the enzyme, since the bonds can be reformed. However, extreme changes in pH can cause enzymes to denature and permanently lose their function. Enzymes in different locations of our body have different optimum pH values. since their environmental conditions may be different. For example, the enzyme pepsin functions based at around pH of 2 and is found in the stomach, which contains hydrochloric acid. Because enzymes are sensitive to changes in acidity, most living systems are highly buffered. That is, they have mechanisms that enable them to maintain a constant acidity. The other factors which can affect enzymes activity are substrate concentration and enzyme concentration. Changing the enzyme and substrate concentrations affect the rate of reaction of an enzyme catalyzed reaction. Controlling these factors in a cell is one way that an organism regulates its enzyme activity and so its metabolism. Changing the concentration of a substance only affects the rate of reaction if it is the limiting factor. If the substrate is limiting factor, changing the substrate will affect the process. If the enzymes are the limiting factors, then changing the enzymes will affect the process. If it is the limiting factor, increasing concentration will increase the rate of reaction up to a point, after which any increase will not affect the rate of reaction. There will be saturation point. Then after saturation point, there is no effect of increasing or decreasing that substance. This is because it will no longer be the limiting factor and another factor will be the limiting factor or limiting the maximum rate of reaction. Substrate concentration. Increasing substrate concentration increases the rate of reaction. This is because more substrate molecules will be colliding with enzyme molecules. As the enzyme's working mechanism is physical, that is the collision between the enzyme and the substrate. When the substrate concentration increases, then the collision will increase so that there will be high reaction or rate of reaction increase. So more product will be formed. However, after a certain concentration, any increase will have no effect on the rate of reaction since substrate concentration will no longer be the limiting factor. The enzymes will effectively become saturated and will be working at their maximum possible rate. Enzyme concentration. Increasing enzyme concentration will increase the rate of reaction as more enzymes will be colliding with substrate molecules. Just the work of enzymes is physical. Therefore, increasing the concentration will increase the collision and increase the reaction rate. However, these two will only have an effect up to a certain concentration where the enzyme concentration is no longer the limiting factor. Everything has its maximum or optimum point. Enzyme inhibitors. Inhibitor means preventers or prevention. The things which prevent the enzyme's activity. Enzyme activity can be inhibited in various ways. Inhibition could be reversible or irreversible. When we say reversible, it has a solution. What are the solution is just by increasing, for example, the substrate concentration. There are two kinds of reversible inhibition, such as competitive and non-competitive. Competitive occurs from its name. There is a competition. Competition between what? Substrate and the inhibitor. Occurs when molecules very similar to the substrate molecules bind to the active site and prevent binding of the actual substrate. Penicillin, for example, is a competitive inhibitor that blocks the active site of an enzyme that many bacteria use to construct their cell walls. So penicillin is a drug which is important to prevent bacterial effect. This is competitive inhibition. This is an enzyme. This one is inhibitor which can bind to this enzyme. So this one is the substrate. This shape, the substrate shape around here, and this the inhibitor shape around here, it is the same. Therefore, it can't fit like the substrate fits with the enzyme. Therefore, it can compete for this active site. Its name is competitive, is because of this. how these non-competitive inhibitors inhibit the enzyme's activity. In some cases of non-competitive inhibition, the inhibitor is thought to bind to the enzyme in such a way as to physically block the normal active site. For example, you can consider someone stands on your passage on the door and prevents you to get in or get out. That is physically blocking one way. Therefore, the inhibitors will come and attach it to the enzyme and then block the right substrate. In other instances, that is, we can consider as case 2, the binding of the inhibitor is believed to change the shape of the enzyme molecule by changing the shape of the enzyme molecule, thereby deforming its active site and preventing it from reaching with its substrate. In this case, allosteric inhibition, we can say. The place where the inhibitor binds to the enzyme is called the allosteric site. And the enzymes mechanism, how the enzymes react or contact with substrates is just physical. All things within the enzymes are physical things. Okay, this is physical. Again, this one is also physical. That is, it is also affecting the shape. Let's see these cases. Frequently, an end product of a metabolic pathway serves as an allosteric inhibitor on an earlier enzyme of the pathway. This inhibition of an enzyme by a product of its pathway is a form of negative feedback. For example, let's say this is a pathway, just making of ATP. You can consider this as a glucose. Finally, there will be ATP. Therefore, this kind of the negative feedback means the final product of this pathway will prevent the earlier enzyme's activity. Therefore, let's say A converted into B by the help of enzyme A, B into C by the help of enzyme. This one is enzyme 1, E1, enzyme 2, E2, then E3, enzyme 3. Finally, there will be ATP after series of steps. The presence of a lot of ATP in cells will prevent the action of the earlier enzyme here. That is, we call negative feedback. The previous cases, case 1, here is the inhibitor. This red inhibitor is physically blocking the substrate. If the inhibitor comes before the substrate, and then it will take this position and then physically block the substrate not to come to this active site. AC means active site. This one is the allosteric site. This is case 1. And this one, in this case, it is not, the inhibitor is not blocking physically, but as it is attached to the allosteric site, it will push the enzyme's structure, and then the enzyme's active site will not be normal. Therefore, the substrate come and cannot fit. Then there is no complementarity. Then there will no enzymes catalysis process. This is non-competitive kind of inhibition. Activators, allosteric control can involve stimulation of enzyme action as well as inhibition. Actually, the non-competitive inhibitors are not all bad. They may stimulate the enzyme's action. An activator molecule can be bound to an allosteric site and induce a reaction at the active site by changing its shape to fit a substrate that could not induce the change by itself. Common activators include hormones and the products of earlier enzymatic reactions. This is the opposite of negative feedback. Allosteric stimulation and inhibition allow production of energy and materials by the cell, that is stimulation, when they are needed, and inhibit production when the supply is adequate, that is inhibition. This is a kind of non-competitive inhibition. Just this inhibitor is not competing for this site because this inhibitor size cannot physically fit with this site. Therefore, it will only attach it to the allosteric site of an enzyme. This site is for active site where the substrate can come and how this reversible inhibition can be reversed by increasing substrate concentration. This is one solution. Then reversible inhibition. Irreversible inhibitors usually covalently modify an enzyme, and inhibition can therefore not be reversed. Okay, there will be covalent modification. The covalent means related with a bond, so there will be bond modification. They do not function by destroying protein structure, but by specifically altering the active site of their target. All the effects, all the processes in the enzyme related things are all physical, related with the active site and the like. When you say target, just substrate. Thank you so much. This is all I have for today. If you have any comment, any question, please write in the comment section under this video. If you haven't subscribed to my channel yet, please subscribe. It will highly encourages me. Bye bye. Have a nice time.